доктора Гига, нет, я все равно неправильно скажу, Кристиан, я никогда не говорю нормально, да? А потом Янина сделает уже более подробно, расскажет об исследованиях, связанных с разработкой новых методов анализа ассоциаций. Okay.
Thank you very much. So thank you very much for a very nice introduction. And also,
could you please speak to the microphone because we have
I tried. Just does it work? Hope so. So as said, thank you very much for the very nice introduction, and also thank you very much for the nice invitation to know the work. For us, it's the first time, but hopefully not the last time. So, my talk had the title From GWAS to Meaning. So you can find out from title it's going about GWAS, genome-wide association study, but not only GWAS, but also on things which can be done after these very nice genetic loci are found in your data or in additional data. We coming from Helmholtz Center in Munich, in Germany, and the first line which you can see here is more or less the motto of the of our center in Munich. So we are in most of the, at least in the part of where we are working, most of the projects are about genes, environmental factors, and common diseases. And in the best case, it's genes together with environmental factors having some influence on common diseases. And the project which we want present you, at least at the end of this, or at the second part of the presentation by Janina, will exactly fit in this motto of our research center in Munich. So it's really in the central of our research things in Munich, which we're doing currently. This slide here is more or less the fundamental things which we have in Munich. So we have one of these very large epidemiological studies in Munich or in Augsburg, which is a city near to Munich, and we in most of our studies use this study which is called KORA study. This means in English, collaborative health research in the region of Augsburg. And as I said, it's a typical epidemiological study, and here are some of these major facts of this KORA study. So the general study of the KORA study is that it's in a certain sense co-financed by many different people, so we can do a lot of phenotyping in this study. We have a lot of information about phenotypes and biosamples from the participants in KORA, and we have a very large biobank, which is very important for us because what we are doing is genetics, and we are looking at biosamples. So this is really the basic of our work in this KORA study. So as resource, KORA is a study where we have more or less 18,000 participants, so relatively big. We have a recruitment age from 25 to 74 years of the participants, and the recruitment of the persons is somewhere between 85 and 2,000 years for the baseline, and then we have some follow-up studies of our KORA study. As I said, we have a lot of information coming from interviews, from questionnaires, from physical examinations, and we have blood and urine samples, and finally from blood we got some DNA samples from each of these participants who are participating in KORA. So, as I said, here's some of these phenotypes. So, like in most of these epidemiological studies, we have a broad range of phenotypes, starting from height, weight, BMI, also things like lung function, for example, going down to things like gene expression information or metabolomics. So information about small molecules. And this will be one of the main resources which we use in our projects. Moreover, we have genomics data. So we have for about 10,000 KORA individuals genome-wide data in a certain sense. So we have for most of the persons, we have up to 2.5 million genotypes per person, which can be measured by this modern genotyping chip technologies. We have metabolomics data, so data about these small molecules. This allows us for about 30,000 KORA individuals to collect metabolome-wide data, meaning that we can, for most of the participants, measure up to 400 metabolites. We have KORA transcriptomics data, and finally we have epigenetics data for most of these participants, for which we also have this metabolomics data. So this is a resource with which we can work, and everything what we show today to you is more or less based on these molecular and genetic data. And I think most of you have sometimes heard about these new form of genetic studies since several years. So this GWAS, genome-wide association studies. And here's a list simply to give you an overview over things which we have done during the last few years, the last 5 years. And it's really, in most of the cases, these studies have been done in cooperation with many other studies worldwide. So again, just some examples. Several blood traits where we were leading, and I will show you an example later. There's a lot of glycemic traits associated to diabetes, for example. Things like obesity, height, urate, lipids, where, for example, URI worked a lot in this area. And again, our metabolomics data, cardiovascular diseases, but also things like restless leg syndrome, which is a very interesting disease, also collected from a colleague at the Harvard Center. Okay, and now this is more or less the state of the art at the moment when you look in these genome-wide association studies. So here, for example, for type 2 diabetes, we have currently the largest study has more or less 35,000 cases and over 100,000 controls, and this 35,000 cases are collected from a lot of different cohorts, including KORA on our own study. In this case, you see, for example, that we have currently more or less about 56 genetic loci which are found in these amount of cases and controls. For example, for glycemic traits, which is a quantitative trait, we have more than 130,000 individuals, where we also could find more or less 53 different genetic loci. little bit less with lung function, for example. For lipids, we have one study which is about which has more than 100,000 individuals, and in this case we could find 95 loci and so on. And there are two studies which I want to explain a little bit more in detail, because these are two studies where we mainly worked on. It's for urate, for uric acid. In this case, we selected about more than 140,000 individuals and found about 30 loci. And metabolomics, here again, in this case with 3,000 individuals, we could find 37 loci. And when you compare these numbers here, you see it seems to be bit more easier to find something in metabolomics compared to compared to lung function or type 2 diabetes. And this comes, at least one reason is that these things like these small molecules are very close to genetics, so it's the power and the statistical power is much higher for metabolomics compared to type 2 diabetes. And so, as last more general slide, when you work in this area, you can see currently there are more than 100 GWAS studies with different phenotypes. A lot of publication in this area. I just counted for us 8 in Nature, more than 14 publications in Nature Genetics, in Science, several publications, diabetes, and so on. And all this work is done, as I already mentioned, in one of these large consortia like Engage, Cardiogram, Diagram, and so on. So this is really very much, pretty much studies where people worldwide work together. Okay, and now I want to just on a few slides give you a little bit an overview of what's coming out of these studies. For example, this is a study coming from the HemGen consortium, where we at the Helmholtz Center, together with the Sanger Institute, had the lead in this study, and we looked at, in this case, on blood cells, blood cell count, and blood cell volume, and these traits are known to be very important for things like diseases like cancer, cardiovascular disease, metabolic diseases, and autoimmune diseases. And we thought in this study it would be good to look at this different blood parameters in common. So we looked at 8 clinically relevant hematologic parameters and tried to analyze them together and not trait by trait. The result, a summary of this paper, of this study, was that we could find almost 70 or 68 traits for platelet number and volume. This was one publication, and in another publication 75 red blood cells in a second publication. And for us in this study, and this was important for us to work further with these results, was that there's a certain enrichment of signals at genes. So when you look at, you usually in this genome-wide association studies, you look at genes, but we saw that many of these SNPs lie very next to or in genes, and then you can do more than only reporting the SNPs, because you can go to databases, for example, and try to find out what's happening with these SNPs and genes, and we saw immediately that many of these SNPs lie close to genes, and these genes are connected via some of these protein-protein networks. And then we thought, oh, it's a cool thing. There's more information than just finding a SNP, and we went with these genes to further experiment, like knockout experiments of mice, or knockdown experiment for Drosophila, or knockout experiments for silverfish, and in this example, we found that many of these loci where we did our animal experiment, we found also severe hematological parameters in these animals. So we had immediately some replication experiments in these model animals. And this is what you want in principle when you look at these genome-wide association studies, because it's no longer so very useful to look only on these SNPs data and reporting some statistical measurements, but you want to make some follow-up experiment where you can really point to very important genes, for example. And now maybe in another few minutes is one of my it's a second second study. It's a GWAS with metabolomics, and this is something which will later be explain to you a little bit more in detail. Just to give you the overview. So for several years we are in Munich working on metabolomics data, and we were worldwide the first, together with others, who conducted a series of genome-wide association studies, and we were quite successful because we could find in these genome-wide association studies for metabolomics relatively large effect sizes for these found loci, and we were able to find a lot of risk loci for metabolites, which are already known to be risk loci for certain diseases. So, for example, there are several loci for diabetes here in this case, or for cardiovascular diseases, like here in this example, some well-known loci for myocardial infarction, for example. And when you look here on one of our cartoons, you can see the areas in which we found these metabolites. Many of them are in lipids, polypeptides, carbohydrates, amino acids, and so on. So we have some transporter proteins. So we have found a lot of relatively interesting loci associated with our with metabolites. And when you now lay over already known disease loci, you see immediately that we have here several loci where we already have known that, for example, this one here is known to be a risk factor for gout. Others are risk factors for diabetes and all these things. So we have immediately some loci where we are quite sure to look closer on these metabolite and genetics associations. Okay, so But it's not only possible with the metabolites to look on its genetics, but you can also look at things like associations to smoking, coffee consumption, type 2 diabetes, gender, alcohol, nutrition, and so on. And we think in these by looking at these metabolites, it's always possible to build up quite a nice picture between what's happening there. Namely, there's a genetic association with metabolites, but also these metabolites are obviously, by definition, more or less associated to very interested things like alcohol consumption, for example. And to build up these whole pictures, it's often very it's very interesting to find these things coming from these analysis. And from this point on, I want to give give the opportunity to Janina to show her things about the association of genetics and metabolites, and to more sophisticated methods she has developed in her work. So, thank you much.
Questions now or after? What do you prefer? To answer questions now or after Janina finish?
At the end.
All right.
So, thank you very much for the introduction, and also thank you very much for the invitation and for the opportunity to present our results here. So, as Christian said, my talk is about genome-wide analysis of metabolite data, and my subtitle is From GWAS to Refined Strategies. And in the next 30 minutes or so, I'm going to present you our basic GWAS and then 2 refined strategies. And the question I would like to ask and hopefully answer is, do we gain from refined analysis? And if yes, what do we gain? So the outline of my presentation is that we have on the top our genome-wide data and our metabolites. So this is the basis for all our analysis. And the straightforward point, if you have the genome-wide data, is just to calculate GWAS. So I'm going to present you 2 different GWAS analyses. One Christian already mentioned, and I'll show you another one. And then 2 refined strategies. So you see there the PSEA, which is phenotype set enrichment. This includes not only the analysis of one metabolite, but of more than one metabolite. And the other one is the non-additive effects analysis, and this is a project which was done here in cooperation with Yuri Alshenko's group, and where this is currently ongoing work. But I would like to present you the first results there. Okay, so first, just briefly, we have our data, our metabolites, and we have metabolites with 2 different technologies. We have even more, but I would like to concentrate here on our most important ones. So we have Biocrates data, which is targeted metabolites. We have there around 150 to 160, depending on which quality control you apply. And this was measured by using electrospray tandem mass spectrometry. The second metabolite technology is Metabolite. And in comparison to Biocrates, this is an untargeted one, meaning that you measure different compounds and you map these compounds after that to known concentration of metabolites. And there are yet some metabolites which you can measure, but at the moment you're not really sure what you really measure. So this is the difference there. And we have there around 250 metabolites. And this is also a mass spectrometry approach, but a little bit different than the Biocrates. As Christian said, we are mainly using KORA as database, and we are here concentrate on one KORA study, so it is a KORA F4 study. And we have for KORA F4 both metabolite panels methods, so Biokratas metabolites for around 3,000 individuals, and the metaboline metabolites for a subset of 1,800 individuals. And at the time point of the analysis I'm going to show you, we had genome-wide data from the FE 6.0 panel, and these were measured for 1,800 individuals, and these are the same as this one here, for which we have the metaboline metabolites. So in the meantime, we have more genotypes measured, but this was the data at the time point of analysis. And we have also the data imputed to HapMap2 imputation. Okay. So for our database. And now I'm coming to the point to show you two different GWAS strategies. So I'm sure that most of you have already heard about GWAS, but just to put everyone on the same page, the GWAS is a genome-wide association analysis, and it has some assumptions. So the first assumption is that the SNP is biallelic, so meaning that we have just three possible genotypes, AA, AB, and BB. And most GWAS also assume an additive effect. So the three genotypes are AA, AB, and BB are coded as 0, 1, and 2. Another assumption is, or another approach is that most GWAS calculate just a regression approach to all of the SNPs on the phenotype. And this can be for, if you have a quantitative phenotype, this is a linear regression approach, and if you have a binary phenotype, this is a logistic regression. As we are doing this for each SNP, of course, we have to correct our significance level, and the usual significance level is 5 times 10 to the minus 8 in this analysis. So, the first study I would like to present you is the Illig et al. Study. It was published in 2010 in Nature Genetics. And it is based on the Biokratas data. So KORA F4 and the Biokratas data I've just introduced you to. There, they analyzed not only the 163 concentration, but also all possible combinations of ratios. So they divided each metabolite by each other metabolites so that they get concentration ratios. And they represent sometimes really interesting enzyme activities, for example. So therefore, they included this also in the analysis. So they had more than 20,000 phenotypes to analyze, all of them genome-wide. As discovery cohort, as I mentioned, it was KORA F4 used with 1,800 individuals, because for these we had both the genome-wide SNP data and the metabolites data. And for the replication, there was the data used from the Twins UK study. And the Twins UK study, as the name said, is a cohort sampled in the UK and consists mainly of twins, of course. And we are using here mostly unrelated. In this study, there was related data used, but it was with a slightly different approach as in KORA. And this is really good replication cohort for KORA, because the metabolites were measured in a really similar way to KORA. So we hope there are not large difference between both cohorts. Therefore, this is good, and you'll see this for other projects also as replication cohort. In this study, there were 500,000 genotyped SNPs used, and their replication and the genome-wide significance level had to be corrected for the 20,000 tests, so it went to 10 to the minus 12. The result of this study is that there were 15 loci found. And in this picture, you see that each of the boxes represent one loci. There is always stated the gene which is nearest to the SNP with the highest p-value, and the metabolite. And you see, perhaps if I move this way, so if you see, all these metabolites except these two are somehow linked into pathways. So they could be really, what the method of is that the result we found are really meaningful, or we can make meaning out of it for the physiological. So, for example, we have here several metabolites which are somehow involved in the fatty acid metabolism, or in the energy metabolism in general. There were 2 metabolites which were somehow involved in the transport of different metabolites, and also here are 2 which are involved in the amino acid metabolism. So of these 50 metabolites, 10 could be replicated. So, and on these data I've shown you, and in the meantime, so after this study was published, we also could replicate the remaining 5 with a larger panel of the Twins UK study. So now all these 15 hits are replicated. The next study was published by Suhre et al. In 2011, and this is very similar approach, but based on the metabolites. So again, they used all concentration and calculated all ratios, and if metabolites has more concentration, of course there were more phenotypes to analyze. Their approach was slightly different. They not used a strict approach of discovery and replication. What they did, they used the KORA F4 and the Twins UK as 2 cohorts, and then screened for promising SNPs. So SNPs which had a low p-value in both cohorts. And then these promising SNPs were then analyzed in a combined analysis, and only these which had in a combined analysis a low p-value were claimed as significant. And of course, the p-value had to be corrected for these tests, so again 10 times to the minus 12 here. The result, what they found in their analysis, you have already seen this plot. So they found 33 loci, and this plot, as Christian told you, is just a visualization of the results, so how these results we found fit into the different pathways. And as Christian said, there are, for example, here you will find again genes and metabolites which are involved in the lipid metabolism, and as it was also found in the bioarchitects analysis, here are, for example, ACADM again. So this was found in both studies. But this, as here are more metabolites included, this also identified more loci. Okay, this was the results from the GWAS. So this was published some years ago, and this was just the straightforward analysis. We have these huge data and calculate linear regression for nearly 50,000 phenotypes. Then, as next, I'm going to show you some refined analysis. So, like post-GWAS analysis. The first is the phenotype set enrichment, and the situation is that we have so many metabolites, and of course they are highly correlated sometimes because they are of very similar chemical structure, but that this correlation structure is just disobeyed by any GWAS where the phenotypes are analyzed separately. So our question was, can we identify more loci if we try to analyze more than one phenotype at a time? So can we somehow exploit the correlation? So which includes information. And therefore we need a new analysis strategy, and we analyzed this with phenotype set enrichment analysis, or shortly PSEA. And this is a strategy for the analysis of multiple phenotypes, and it is gene-based. The specialty about PSEA is that it analyzes phenotypes in sets. And a set is just a set of phenotype with no more recommendations, no more assumptions. So it can be made up by prior knowledge, by experiments, or just by random, or with a p-value criterion. So you're really free in defining these phenotype sets. It uses, as the name said, enrichment methods for the identification of enriched phenotype sets. And this was also published in Genetic Epidemiology in 2011. So I would like to really briefly show you what is the method in a nutshell. So our starting point is, as you can see here in the upper left corner, we have SNP-based test statistics for a panel of phenotypes. So, and in this graph, I'm using different shapes for different phenotypes and different colors for different genes. And the strengths of the color represent the strength of the association. So, as I said before, it is gene-based. So, our first approach is coming from these SNP-based test statistics that could, for example, be a GWAS results with just chi-square test statistics. From these SNP-based test statistics, we must now go to the gene-based test statistics. And we did this just by picking up the strongest association. So for each phenotype and each gene, we took this SNP which had the strongest association. With this, we have our gene-based test statistics. The second step then is to calculate the enrichment score. And there we have on the right hand, again, our gene-based test statistics from the first step. And on the left-hand side and on the right-hand side, we have our previously defined phenotype sets, which are made of some knowledge or just randomly. And to calculate the enrichment score, we just then sum up for each gene the gene-based test statistics of all phenotypes as a set. So, for example, you have here the enrichment score of gene 1 and phenotype set 1. And then we analyze, because the phenotype set 1 is made up of phenotype 1 and phenotype 2, and we then sum up the gene-based test statistics for the two phenotypes. And we did this just for the whole genome. Of course, now we have to know if this enrichment score is somehow, yes, if it's really higher than we would expect it randomly. And therefore, step 3 and step 4 are the permutation and the estimation part. What we did, we had our large matrix where we have the individuals in the rows and the phenotypes in the columns, and we then shifted the column of this matrix. So the correlation structure of the phenotype stays the same, but the assignment to the individuals is destroyed. And we kept the genotypes as they are and generated with this random phenotypes which have the same correlation structure as our real phenotypes. And with this, we then can calculate it again. Oh, there's an error missing, so here's an error. We calculated then GWAS and have here SNP-based test statistics for our random phenotypes, and could with this then again start with step 1 and 2 and calculate so our permutated enrichment scores. The testing is then, we did this several times, so in the analysis I'm going to show you it is 10,000 times, and the p-value then just counts how often the enrichment score of the permutated phenotypes is higher than our enrichment score for our phenotypes, and that divided by the number of permutation is our p-value. Of course, you can imagine that the calculation of GWAS for all phenotypes and for all permutation is really computer intensive, so therefore we had to limit this to 10,000. This is the strategy just in a nutshell. And there, as I said before, we had to define the phenotype sets somehow. And we decided to define them on single association, so on the association of each gene, of each gene to a single phenotype, and applied their p-value criterion. So this is when you have here your phenotypes, for example here 5, and you have the minus log 10 of the p-value on the y-axis. You then just apply a significance level which is fixed, and took all phenotypes which had a p-value below this level as elements of the phenotype sets. And this is done for all genes. So at each gene you can identify at maximum one phenotype set, and then these phenotypes are tested with the strategy I've just shown you before. This is the method, and of course we applied it to data. So we applied it to the metabolites. We applied it also to the Bayard-Cortez metabolites, but I want to show you the results for the metabolites. And we applied it to 193 metabolites. And okay, again, KORA as discovery and 20UK as replication cohort. And the result is that we found 23 loci at which at least one genotype set was enriched. And for these 23 loci, 17 were already known from GWAS on the same data. So as, for example, in the Suhre et al. Paper I just showed you before, and also by other published GWAS on large metabolite data, but they all have the similar number of individuals. So compared to GWAS with similar number of individuals, we identify 6 loci which were not published in these ones. And these 6 loci are presented here. So you see here are the gene names, and these are the 6 gene groups. Sometimes we have more than one gene, but there are overlapping in some SNPs because we had to do the mapping from SNPs to genes, and there were some SNPs which were mapped to 2 genes, and therefore these genes which have no reticle space between them are in one gene group. And each box in these plots represent one metabolite colored according to their super pathway. There. And I would like now especially point out these 2 findings. So the first is the cytochrome family P454, and this was for this there was a set enriched which contains of lipids, amino acids, but also peptide and a cofactor, and the cofactor was heme. And this family is known to be a family of enzymes which are involved in various processes, for example, the lipid synthesis. And it is also known that heme is a cofactor of this. So this finding was really interesting because our metabolites which we found to be, or the metabolite set which we found to be enriched, contain nearly of substrate and products and also of a cofactor of the gene function. So this somehow makes sense. So this was a really nice finding, and the other finding was, or one of the other findings was LINGO2, and LINGO2 was associated, for LINGO2 there was a gene set which contained mainly of lipids and amino acids, and the gene function for this gene is not known yet, but a large GWAS found to be associated with BMI. So we do not know what the gene makes or what the gene function is, but the enrichment of this set of different metabolites which are associated with the metabolism of lipids may suggest that it could have something to do with their lipid metabolism, and this would also fit to the association with BMI. Okay, so far the results from the first refined strategy. So this was my second part. And now I'm coming to the analysis of non-additive effects, so our second analysis of our second refined analysis strategy. And the results I'm going to present here were mainly produced by Yakov Zabilov here from this institute. So what was the situation? The most GWAS analyzed, as I said, additive effects. So an additive effect assumes that when you have a quantitative phenotype, the increase or the decrease of the phenotype per genotype is equally distributed. So there's decrease from, or the increase from AA to AB is the same as the increase from AA to BB. And our question was, can we identify different loci if we analyze other genetic effect models? Because the additive effect is one effect, but there are also others, like the dominant model, the recessive model, and the overdominant model. The additive model, I already told you that it is the genotypes are coded 0, 1, 2. For the dominant model, it is coded 0, 1, 1. For the recessive model, 0, 0, 1. And for the overdominant model, 0, 1, 0. In this way, we could analyze all these 4 models with linear regression approach as we encode our genotypes in this way. But there's also a 5th gene model, and this could look like this. So here you say that looks somehow like an overdominant model, but the AA and the BB genotype are not on the same level. So this is not really, so the overdominant model does not really fit to this. And this can be analyzed with a 2-degree of freedom test. So if you code the two alleles in two different variables. So you have here the SNP code, the allele A coded as 01 and the allele B coded as 01, and you just include both variables into your model. And of course, this also covers the 4 models I just presented you. So this can be used as screening tool, because if there is an additive effect, this model should also find something. And this is what we did. We used this genotypic model as a screening tool to analyze all our metabolites and SNPs. So we applied it. I just want to say to which data we applied it. We applied it to the same data as in the Illig et al. Paper. So the first paper I presented to you, there was the Biocrates data, and the same is put here in quotation marks, because there was in the meantime a little bit of different quality control, so it was not exactly the same, but still the same measurements. And we looked on all concentrations and all ratios. As again, discovery cohort KORA F4, replication cohort 20OK. And we analyzed 700,000 SNPs, and in the top hit regions we analyzed also the imputed SNPs. So, as I said, the discovery was done in the following way. We screened all SNPs, all metabolites, all concentrate, or all SNPs for all metabolites in all concentration with the 2DF test. And then we pre-selected SNPs which had a promising p-value and good quality concerning special quality criteria. And for these promising SNPs, we then analyzed also the imputed SNPs in this region. And p-value genome-wide significance level was set to 2.1 times 10 to the minus 12. Replication in an analogous way, so 2DF test for all the significant findings. We tried to take really the same SNP, and if this was not possible, we tried to find a SNP which is a really high LD. And we corrected their significance level for all the number of our findings. So at this analysis is still ongoing. I can just show you here some first results. So, but one interesting thing was, for example, this SNP. Here you have the concentration of the metabolites, and these are the box plots for all the 3 genotypes, so AA, AB, BB, and you can really see that this looks more like an overdominant model. So, and indeed, the overdominant model had here the lowest p-value, and this SNP could not be found if we had just applied an additive effects model. So here, the application of the overdominant model really brings us to a new loci, or to a new SNP, so to say. It might be in a loci. And the second example I would like to show you is this one. This is another SNP, and here you can really nicely see that the model of this SNP seemed to be dominant. And indeed, the dominant model had here the strongest p-value, and this SNP could also not be found with the additive effects model. So, but as I said, this is still ongoing, and this is still under work, and therefore I cannot here go into details. We are still working on this. I would now like to summarize. So, what I presented you is that with GWAS we identified several loci with metabolites which are really meaningful in the sense of that it very often lies on a pathway. One main advantage of the GWAS is that it's very simple to apply. So if you calculate it for 20,000 metabolites or ratios, it's more or less meaning of computation time, but it is easy. But the drawback is that it has many simplifications, or simplifying assumptions. With PSEA, I've shown you that the analysis of multiple phenotypes can lead us to new loci which are not found in GWAS on the same number of individuals. And with the non-additive effects model, we have seen that it helps us to understand really the genetic structure behind the effect to the metabolite, and also leads us to new loci, or can lead us to new loci. So to conclude, GWAS are a really good tool, but only a first step. So the refined analysis can really increase our knowledge in gaining new loci or in improving our knowledge on the metabolites. And as future plans, we think of application this to more samples, and we also think about application this to unknown metabolites. And one further project of our cooperation with Helmholtz and Russian Institute is the analysis of gene-gene or gene and environment interaction, also in this setup of data. So with this, I want to close my talk. I hope I could give you a good overview about our GWAS analysis and the refined strategies, and I could answer a little bit the question if refined analysis does help us, and we see it helps at least, and we'll find something new. Before I stop, I would like to acknowledge a few people. So especially Christian Giger and Konstantin Strauch from the Institute of Genetic Epidemiology at the Helmholtz Center in Munich. Then Soyu Shin and Nicole Soranzo from the Wellcome Trust Sanger Institute in Kingston for providing data of the Trans UK study on doing analysis there. And last but not least, Yuri Alchenko, Yakov Chepilov, and Sotpo Sharapov from the Institute of Cytology and Genetics here at the Siberian branch of the Russian Academy of Science. And especially Yakov Chepilov, who did most of the analysis of their non-additive effects model. With this, I want to stop. I thank you for your attention, and now I'm happy to take your questions.
Please.
So in general, the data is not publicly available to everyone, but we have a structure of project agreement. And if you're interested in the data, you can make a formal request to our institute. I hope I'm saying the correct things. Oh yeah. So we have there the contact data on our internet homepage, or you can. So if you're interested in using it, and we are always happy for collaborations. So.
No questions?
Have you any idea about explanation of biochemical mechanism of dominance? Because if you have protein with A amino acid and B amino acid, YAB combination will be more or less effective than one.
Yeah, that's a really good question. So we had already a lot of discussion about that. So in general, you're right. You would think that the additive model is just the natural way for such biological compounds. But I have learned that there are some examples where the dominant or even the over-dominant model makes sense. So, but you're right. These examples are rare.
Perhaps you said that I didn't hear it. The problem with all this metabolite and concentration, it's very, flexible, I don't know, variable. So how many times you measure the same person, and it's
So for the Biocrates metabolites, in general, we measure each person just one time.
Yes.
There might be two answers to your question. So the first one is to the experimental variance, and for the Biocrates data, we had some individuals which were measured several times, and there we had something like an experimental variance, and we excluded individuals which had or metabolites which showed not to be reliable. And so this was just a quote. So I'm not sure. I think this was around perhaps 5 metabolites, which seemed to be not stable for repeated measurements. And the second answer to your question might be, so we analyzed not most, but 800 of our individuals to 2 different time points, and this is more done for longitudinal data.
Just more about stability, like you said about all these hematological traits. I mean, it depends upon, time of the day, what you eat today, and so on. And you just, well...
But one thing we didn't mention is that we usually do our analysis only with fasting samples, so everyone in the study is fasting.
Yes, that's all right, but still, what we are measuring.
Yeah, we made one experiment where we looked at our measurement, and we saw that this metabolite per person is surprisingly stable.
So that was verified. I mean,
this is verified.
So, right. Anyone have questions? All right, then we have more. Okay. Yes. First, I don't understand what twins has to do with all this approach. I mean, that's. I mean, I don't think. Okay, you explain.
The study is just called Twins UK, and it was sampled from twins, but we are not using the fact that they are twins. So
right, you ignore this.
We ignore this, and monozygotic twins there's one excluded from the analysis. This study has just the advantage that the biochar metabolism and the metabolites are really similar to KORA.
Right, then another question. No more questions from the audience? All right. Well, that's very general questions. Question. Well, you put this value, well, statistical 10 and minus 25 or something like that, 12. So my question is, you are just trying to find smaller and smaller effects. Does it make clinical sense? So how strong effect should be to consider it clinically? Do you have any idea in this? All right, they are significant, but does it make change in my life or whatever? And the strategy of the therapy or whatever? Do you have any sort of, how to say, commercial level, the significance level of the effect? From this effect you can ignore it. It's all right, it's significant, but you can ignore it in your life or whatever. Any ideas?
So
there's obviously more than one answer. So we are in the range of any other biomarker. So I'm quite sure that any other biomarker which is can measure in the body is more or less on the same range like we have here. We currently do not really look at things like finding extremely good biomarkers. But you are right, this is really an important question which we have to answer. I personally don't think you will find one of the metabolites, and this one metabolite will be the best biomarker, whatever. We are looking more or less for profiles of biomarkers and all these things. And I think that it is possible to find a group of biomarkers which play together and have a certain consequence in clinical science. But I don't think that we really don't currently something which is for sure the best biomarker. But it's
all right. All right. That's another question I always ask Yuri to do, because you always there is a sex dimorphism, everything. But what you do, you just adjust it for sex and happy about that, and then it goes in your computers nicely, and that's all. So what I always asking him to do is to find what they call the sexually antagonistic traits. which is good for males and bad for females and vice versa. There should be something, but have you done anything in this direction, or, well, in general, anybody else?
So we have done an analysis for analyzing if there are specific differences in the effects on the metabolites.
Yes.
So I think we found 2 or 3 metabolites where the effects of in men and males and females were significantly different. So, but this is not the complete answer to your question. So you would like to have a positive and a negative effect.
Well, the nature, so they are under different pressure of natural selection in males and females.
I think our effects were in the same direction, but just the female one was bigger, I think, than the male one.
Do you know anything from literature in this sort?
I don't know. So
model?
At that time that we wrote this paper, there was nothing about this. So I think uric acid might be your example, Christoph.
Right. My last question. That's my last question. You were speaking about non-additive effect in between alleles of the same gene, but as I understand, in this genome-wide, you also suppose that there is sort of additivity between loci. So each loci contributes its separate, its separate contribution. It doesn't interfere with others. Are there any approach to to reveal these interactions and all that, or it's hopeless between loci interaction?
Yeah. So this was in our future plans. We think about the gene-gene interaction.
Any idea how to do it? No,
we are still discussing it.
But so you will not going to tell me that. All right.
No, we have just no solution for it yet.
So there is no risk I would understand, but anyway. So, if no more questions, thank you very much.